All right, this is going to be the second video for the Module 15, the urinary system. Okay, we're going to pick up on page 496 at urine formation, step 2, reabsorption. All right, reabsorption occurs once the filtrate enters the proximal tubule. So remember, it's gone through the Bowman's capsule and it's gone through the glomerulus. Reabsorption, now you've got the capillaries that are wrapped around the nephron. So it occurs once that filtrate enters that proximal tubule, the one that looks like the heart, the one closest to the glomerulus. So once it enters there, that is where the reabsorption process starts. Remember, filtrate is both the useful and the excess or waste molecules. All of that stuff is in there. It dumps everything out, and then it's going to reabsorb the useful things for the body. So the blood has to reabsorb useful molecules and leave the other stuff in the filtrate to be disposed of in the urine. So figure 15.3. Remember those efferent arterioles that we looked at? So the efferent and the afferent. The smaller one is the efferent, and it takes place by the efferent arterioles that leave the glomerulus and are wrapping around the nephron. This is called the peritubular, because it's so rad, dude, the peritubular capillaries. So you have a good network that are surrounding the nephron. As the filtrate moves through the nephron, the substances that the blood need are going to pass across the wall of the tubule and back into the capillaries. This is reabsorption. Pass across the wall of the tubule and back into the capillaries. Reabsorption. The filtered molecules are reabsorbed here. The walls of the nephron tubule are really thin, so reabsorption is really efficient. Also, you have a lot of surface area to increase the absorption rate. So just like in your small intestine, lots of surface area here. So you have these things called a brush border that's like bristles on the inside to help increase the surface area so there's more absorption. Most of the reabsorption happens immediately in the proximal tubule, the heart. About 65% of your water, 100% of the glucose that your blood needs is going to be reabsorbed here. Once the filtrate passes through the loop of Henle, on some of them, remember only 10% have a loop of Henle. So once it passes through the loop of Henle and then the distal tubule, the one further away, it enters the collecting duct and as urine is your final product there. So absorption occurs in two ways. You have active reabsorption, and that requires active, right? It requires ATP and a carrier. It's bigger. Passive reabsorption, it doesn't need any cellular energy, so no ATP is needed. So active reabsorption. Let's see. Active reabsorption. All of your nutrients and your minerals that your body needs are absorbed in this way. So glucose, amino acids, vitamin C and B, sodium ions, potassium ions, calcium, any of those things your body needs are going to be through active reabsorption. And they're a little bit bigger. They're going to need energy to be absorbed. So they must have a carrier to take them across the nephron epithelium, and it takes energy for that to happen. The exception to the carrier, yay, are these leaked proteins that happen to make it through that don't need a carrier. Some of them leak through and they don't need a carrier. These cells are taken in by pinocytosis. You remember what pinio is? That's cell drinking. So they're going to be dissolved in that fluid around the cell. It's going to fold in and pinch off that protein and pull it in. So most of them need a carrier. Some of them get to move across through this pinocytosis, the cell drinking. Those that need a carrier, an important effect takes place, and it's called the tubular maximum, or the Tmax. You need to know what that is. Tubular maximum, or Tmax. This is the maximum rate of reabsorption by active transport through the nephron tubules. This limits how much of a substance can be reabsorbed. So Glucose is going to show up in your urine if your blood glucose is too high, because it's dumping the extra glucose in your body out through your urine because it didn't need to reabsorb any more of it. Remember, 100% of it got reabsorbed there. So if it didn't need it, it dumps it and it goes out in your urine. So one of the quickest and easiest and cheapest ways to check to see if you're having problems with a diabetic's having trouble is to test the urine for glucose. So glucose, a big way for it to do that, has to have this carrier. Sodium normally is in your urine because of the diet, because of our diet has so much sodium in it. So the Tmax for sodium is always reached, and we have sodium in our urine. It's a way that our kidneys are going to control whether or not something gets reabsorbed, is the Tmax. So know this, if the Tmax of a substance is low, then the kidney is designed to limit the amount of it. If the Tmax of the kidney is high, then the kidney can allow for higher concentrations of that substance in the blood. Okay. All right. So that's active reabsorption. It requires energy. It has to have a carrier to move the things out, or through pinocytosis, but it still requires energy. Now, passive reabsorption, remember, no ATP is required. No energy is required. So there's no limit on what can be absorbed in this way because it doesn't require energy. So this is how water gets reabsorbed, because it doesn't need a carrier, because water is just so tiny. It doesn't have to have that. Osmotic pressure also attracts water back into the capillaries as well. So there are a lot of proteins that couldn't leave the capillaries, and now there are a lot of reabsorbed nutrients. So osmosis is caused by these molecules that are drawing the water back into the capillaries. So that blood is really dense and full of nutrients now, and the water really wants to go there because there's not a whole lot there. It wants to get out of where it's crowded and get into where it's not. So the problem is that waste products that are left there get even more concentrated. And if the waste molecule is small enough, it can diffuse back into the blood as well. So more of that higher concentration to a lower concentration area wants to get away. We don't need the waste products. We don't want those things. An example of this is urea. It will flow back in because it's really small. So some of it will always be in the blood, but most of it, most does end up back in the blood, in the urine. All right, urine formation, step number 3, secretion. You have waste products that didn't get filtered that are specifically sent into the nephron. So it's picking out those things that were left to dispose of, the staples and the little pieces of paper and the things like that. So it happens mostly in your distal tubule, the one that's further away from the glomerulus. More gets reabsorbed than what actually got left behind to secrete. So most of your potassium and hydrogen are secreted here. Remember, hydrogen is what's controlling your pH. So the ability to donate hydrogen makes a molecule an acid. So if the hydrogen gets added to H2O, it makes it more acidic. So hydrogen taken away makes it more basic. So if the pH in the blood starts to get low, then hydrogen is going to be secreted into the nephron so that the blood pH increases. If the pH of the blood is too high, no hydrogen will be secreted. All right, so urine formation, step 4. We've got the reabsorption of the water. So secretion happens from capillary to, that's a little too light, capillary to nephron. Secretion goes this way. Okay. You got your capillary here and your nephron here, and secretion is going to go from capillary to nephron, secretion. So reabsorption of the water, now you have an amount of water that gets reabsorbed into the kidneys, and it must be really tightly controlled. If you drink a lot of fluid, the kidneys have to get rid of the excess. But if you're low on fluid, then they need to conserve the amount of water that you have there and not let too much out into the urine. They need to regulate how much water is being reabsorbed and how much water is being released. Normal interstitial fluid has a solute concentration of about 300 milliosmoles per kilogram. The interstitial fluid that is deep in your kidneys is different from this. So blood is about 300 milliosmoles per kilogram as well. And the interstitial fluid in the kidneys cortex is also about the same. So deeper though into the medulla, it reaches about 1200 milliosmoles because you have urea. That's where the urea is. And it's in higher concentrations. So figure 15.5 shows you a really good picture of a nephron and the vasa recta. So this is how water reabsorption is controlled. Remember, only 10% of nephrons have a loop of Henle. That is going to concentrate the urine through reabsorption of H2O. The renal corpuscle and the proximal tubule are in the cortex. Remember, turn that thing on the side. The renal corpuscle and the proximal tubule are in the cortex. The outer portion of the kidney, that's where the loop of Henle is going to go down deep into the medulla, where its concentration of the solutes is about 1200 milliosmoles. And then it rises again back into the cortex, and the collecting duct goes through into the medulla, into the minor calyx. So the concentration is steadily increasing from the cortex to the medulla. So it's going to get higher and higher when it leaves the cortex and goes into the medulla. Now let's look at how water is reabsorbed. So the descending loop of Henle, so that's the portion that's going down, that descending loop of Henle, is permeable to water and solutes. So the interstitial fluid is about 300 milliosmoles when it enters. As it travels down to the medulla, the interstitial fluid increases in the solute concentration, and this pulls water out of the loop of Henle by osmosis and pulls the solutes into the loop of Henle by diffusion, because it is becoming more concentrated as it's traveling down. So at the bottom of the loop, the water solute here is about 1200 milliosmoles, which is the same as that surrounding medullary interstitial fluid, that fluid that's there in the medulla. So it's highly concentrated at that point. The water that was in the loop of Henle left the nephron and entered, I hope you're following along on your figure 15.5. Please do that. So the water that was in the loop of Henle left the nephron and entered the interstitial fluid there in the surrounding area of the medulla, but it quickly went back to the vasa recta. Okay, the vasa recta is that blood vessel. So the blood reabsorbed the H2O. The blood is reabsorbing the water that left the nephron, which is returned to the body, and it will not be turned to urine. So that vasa recta reabsorbed that water that the body is going to keep. It's not going to be turned to urine. So here, reabsorption goes back from the nephron to the capillary. So, right, secretion went from the capillary to the nephron. Reabsorption goes back from the nephron back into the capillary. So it is going to reabsorb any type of H2O that the body needs to retain. So this thick segment of the ascending loop of Henle is not that thick segment. You can see as the loop of Henle goes down and it starts to come back up, it has a thicker section there at the top. This thick segment of the ascending loop is not permeable to water or solutes at all. Okay, active transport is the only way for something to get out. As the filtrate travels up that thick ascending portion, only three things leave the nephron. Sodium ions, potassium ions, and chloride ions. All of those have a carrier molecule in that next portion of the loop, and they can be actively transported out and back into the interstitial fluid and then diffuse into the vasa recta. So they're going to go back into the blood. Okay, the only way that they can get out is through that carrier back into the blood. All right, the concentration of the solute goes way down at this point in this step, right? Because it's reabsorbing all of that water. So most often water is going to follow the solute by osmosis. It wants to be there because it was highly concentrated. By osmosis, it wants to follow it. So if they cross, right, the solutes cross, then the water wants to follow. In that thick ascending loop of Henle, water cannot follow. A few things are allowed out that have a carrier protein. Nothing is allowed in. It cannot follow. So the sodium, potassium, and chloride ions can go out. So the fluid loses some of that solutes, but the water is going to remain. So the concentration decreases to about 100 milliosmoles there. Urea is the one main thing that gets left here. All right, electrolytes, your sodium, potassium, and chloride ions. When they are dissolved in water, that H2O, is going to conduct electricity. So when they get dissolved into water, that water will conduct electricity. Electrolytes, that's why they're called electrolytes, electricity. Electrolytes tend to follow the water. They're going to follow the H2O. They want to be in there. But the kidney has your kidneys have a special design that is going to retain the water so that it can't stay together. So we can lose water without having to lose our electrolytes, right? So electrolytes are removed from the water and then returned to the blood, and the water is going to stay there in the nephron. So the filtrate now is in the distal tubule. So it's made it all the way around. It's in that distal tubule, the one that's further away, and the solute concentration is really low. It's about 100 milliosmoles. It's less than the interstitial fluid or even the blood because those electrolytes have been transported out of the nephron and the water stayed, the H2O stayed. Next, we'll need ADH, or antidiuretic hormone. We've talked about this before in another module. Antidiuretic hormone is made in your posterior pituitary, and it's controlled by your hypothalamus. So it stimulates by our cells shrinking slightly when we get dehydrated. When we're dehydrated and our nice plump cells start to shrink just a little bit because they don't have enough water, it triggers your hypothalamus to trigger the posterior pituitary to start making antidiuretic hormone. So the antidiuretic hormone is going to control the amount of urine that gets produced. When the blood has a high amount of water in it, then the antidiuretic hormone production is limited and lots of urine gets made. It's going to just keep letting you make the urine. But when you have, when the blood has a low amount of water in it, H2O in it, the amount of antidiuretic hormone released increases so that less urine is made. It wants you anti-diuresing. It wants you not to diurese, not to make urine. It wants you to hold on to it so that your body can fill in, rehydrate your cells. So here's what you need to know. Antidiuretic hormone makes the distal tubule, that tubule that's further away, it makes the distal tubule and the collecting duct more permeable to water. So it makes antidiuretic hormone makes the distal tubule and the collecting duct permeable to water. It wants to be reabsorbing that water so that your body can hold on to it. Antidiuretic, anti-diuresing, not diuresing, not making urine or not releasing urine, not letting the water go out into the urine. It wants you to hang on to it. So the more antidiuretic hormone is present, the more permeable to water those two things are, the distal tubule and the collecting duct. So lots of antidiuretic hormone, then the water is going to move out of the distal tubule and the collecting ducts by osmosis because the solute concentration is really low. You're dehydrated. So it is drawn to the higher concentration on that H2O is going to be going out if it goes into the interstitial fluid and then into the blood. So antidiuretic hormone levels, if you have a high antidiuretic hormone levels, the water is going to leave the nephron, H2O is going to leave the nephron at the distal tubule in the collecting duct and re-enter the blood to conserve water. So not a lot of H2O is going into your urine now, but plenty of waste solutes, so your urine will be concentrated and dark usually. If you have too much water, too much H2O in your body, it's going to decrease the amount of antidiuretic produced, which will decrease the permeability of that distal tubule and the collecting duct to water. So as it filtrates, water may want to leave, but it can't because it's not permeable if the antidiuretic is absent. So it's going to stay in the nephron and then it's going to be excreted by the urine. So if you have plenty of H2O, the distal tubule and the collecting duct is not as permeable to H2O and the urine is dilute. All right, storage and release of urine. Now that you made urine, you have to store it and get rid of it. So remember, your bladder is really stretchy. It's only about as big as your thumb when it's empty, and it can get pretty big when full depending on your own anatomy, but it should hold about a ½ liter of urine. When it stretches, information gets sent to your spinal cord, and emptying your bladder, right, is a housekeeping duty. So the parasympathetic division of the autonomic nervous system is going to kick in. When it gets stretched and needs emptying, the parasympathetic division is going to send impulses to contract that smooth muscle of your bladder, and it's going to empty through the urethra. All automatic, right? So babies do it because a reflex tells them to. So as they age, right, they're developing that myelination of their nerves so that the spinal cord sends a message to the brain and the brain decides what to do with it. Remember, that's one of the last things that gets the myelin sheath wrapped around it, which is why that's one of the last things that you do with a baby is potty train. So that gradual, it takes time for that to develop. So it's just a reflex up until a certain point that they can start to control it. The external urethra sphincter, that's the ring of skeletal muscles that go around the external end of the urethra. It's normally contracted and the urethra stays closed, even if your bladder contracts. Your bladder could be saying, I'm really full, I gotta go, I'm really full, I gotta go, but that external urethra sphincter is going to stay closed. So to empty your bladder, right, requires a conscious effort for you to relax that sphincter. Incontinence is the loss of control of that external urinary sphincter. So if that loosens any, or if you just lose control of it altogether, right, you have lost that sensation. That's what we call incontinence, where you just you pee on yourself. So the nervous system damage, or even in the elderly with that degeneration of the nervous system. Okay, that is the end of our second section. I will wrap it up with one more section of lecture for you to listen to for this module. All right.